Okay, we're back again. This time it's for lesson 2. Applying genetics problems. I think this is the right one. Yep. Okay, looks good. All right, so if we go to question 1, the O blood group, essentially what this question is asking for is if a child has such and such phenotype, and the mom and the dad also have a particular phenotype, what are the actual genotypes for them? And so, like if we come here to A, it's just really not really a guess and check, but you just kind of figure it out. So you see the child has O blood type. So we know that is going to be II. And then we can figure that out. Okay, well, that means that both mom and dad have to have an I in there. And if we come to dad, he's A type, so his blood will be AI. And then for the mom, who also has O type, it's going to be II. If we come to the Rh factor, we have a negative, which means 2 recessive genes. For the baby, the mom also has Rh minus, so 2 recessives for her. And then the dad is a positive, which means that he has to have 1 dominant R allele. The question is, what is his last allele going to be? But we can notice because the child is negative, that means the dad has to at least be a carrier for the negative, and so he gets the R. And for the MN focus or locus, we see that the child has an M, and the dad has an MN. So we can type that in, or sorry, the mom has the MN, and the dad also has an M. So we don't really know, we just know that then it's going to be MM for both the child and dad because they don't have the N carrier in there involved. So let's go down to number 2, which is section B. A proportion of children born to a man with such and such a genotype and a woman with the genotype will have blood type B, Rh minus, and MN. So you just have to find out the specific genotype probabilities for each one and then just times them all through. So first one, we're looking for B blood type. So we come here and we see we have negative. So this one will have no effect on what the blood type is. And we come here and I'm sorry, excuse me, it's A and B. I misread that. This one has a null, or not a null, but an I. So this first one's going to have to be a B because as we look at the dad and the mom, only the dad has a B. So that means that we have a 50% chance of dad giving a B. And then between the two options mom has to give, if she gives an A, the blood type will be AB and won't work. So that makes it only a quarter chance that that the B can be given. Then for Rh minus, we have heterozygous and heterozygous, which means it's going to be a quarter of a chance for the standard probability. So that's another quarter. And then we're looking for MN. So dad has MN. Okay, we need to see what mom has. Mom has NN, so mom is going to give N, which means we need to know what's the odds that dad will give an M. Well, it's going to be 50-50, so then a half chance for the MN. You times everything through, and you're going to get 1 out of 32, because 4 times 4 is 16, times 2 is 32. So 1 in 32 is going to be the proportion or odds. You could do percent 3.02 or something like that. Go down to the next question. Same thing, just different variables. So we need to look for blood type. Only mom has the A blood type. So she has to give A because dad can't give that. And then dad is going to have to not give that B. So we're going to have a quarter chance of that blood type being passed on. We want Rh positive, which means we need at least one of these dominant Rs to be passed on. So it doesn't really matter which one we give. It can be from mom or dad or both of them. And so if you go through the Punnett square, you're going to get 3 out of the 4 squares where there is a capital R put in there. So we get a chance of 3/4. And oops, I'll make that one less space. And then if we go to the last one, we want an M. So it means we don't want N. We have to get 2 M's from both mom and dad. And so that's going to be only a fourth of a chance. And if you times all that through, 4 by 4 by 4, you get 4, 16, 64. And then 1 by 1 by 3, you're going to get 3. So 3 out of 64 is going to give that ratio. In fact, I mistyped that. Yeah, we're looking for A. And then, oh, I think I accidentally copied the dads down. And then M. No, those ratios are right. I did them again as I was talking. So then, so this one's asking, given these two genotypes for the parents, could a specific child receive those? So the dad says, I have B type, mom has O. So because mom has O, whatever the dad has is going to really determine what we can give. Or sorry. Oh, these are, I wish these are rearranged differently because what you see is the man has a B, mom has an A. And so with that, they're both going to have a B and an I and an A and an I. So, or they could potentially have that. And so dad could give an I, mom could give an A, and that would give us the potential, or no, that would give us the potential for A, B. But if they both give the I's, then we would have a O-type blood. Then for RH, both mom and dad are dominant with the positive, but they could be heterozygous, which would mean that there is a 1 in 4 chance that the child could be negative. And then we have MN and N for the mom and the dad. So dad is going to give an N, and mom has the ability to give an M. So the solution is, which I wrote down here, is that this child could be the dad's son, but we don't know just from this. We can't rule him out, though. On to question 2. This is talking about the walnut cones. Okay, yeah, this one. So what the question is, what are the genomes for the parents going to have to be to get a ratio of given ratio? So, for instance, this first one, we need 1, 1, 1, 1. And the only one I found that would work is this one here, capital RR, capital PP, and RRPP. So to get the walnut, which you need both the dominant ones, you would get that dominant, and then the rest wouldn't matter. You'd get walnut. For rose, you would get the rose, but then you would want recessive P, and then you get another recessive P, vice versa with the P there, and then you would want both recessives from this parent, and that would give you the single. And we're coming down to the rose and single. So because there are no P or walnut options, we know that all the P's have to be recessive, so they're all going to be little P's. And then for the 3-to-1 ratio, in a normal S square, you need the heterozygous, where getting an R 3 different ways could give that to you. Rose and single, so we need a 1-to-1 ratio. If we make everything little P's, because we don't have any P's or walnuts to deal with, and then we come here, we know that this parent will give us an R no matter what, and so it's just a 50-50 chance on what this parent gives us, whether a dominant or recessive rose allele. And so that'll give us the 50-50 or 1-to-1. For this one, the 3 walnut and 1 rose, I found a little bit more options. So we know that regardless, there has to be at least one parent with double dominant alleles to pass on that rose, because the rose has to be there. We do need to leave options for the P's because there is no P option. So we need to at least give them little, but we can also leave the dominant in there. And then basically I just found out that as long as one of the parents has the double R's, that it really doesn't matter what the other one has, because then that'll make sure you get at least the rose or the walnut. But yeah, you can't really know for certain which it is. So That's why I have the double uppercase or double not recessive, dominant, and then a dominant recessive, and then double recessive. Coming down to number 3, this is the incomplete dominance. So it's one in the example the book or the readings gave, which was a red flower, white flower, and then in the middle a pink flower. You can break this up. So what I did is, the reason I knew this was an incomplete dominance, and I talk about it here, is like here you get Palomino, Palomino, you get some other things, and you go, well, maybe, whatever. It's hard to tell, but the one I said, yep, this is an incomplete dominance, is, I think it was this one, yep, the chestnut and cremello, because you're taking 2 other phenotypes and you're getting only one of a completely another one. That screamed to me of incomplete dominance. So in this case, I thought, well, there's probably dominant, dominant, and then recessive, recessive, which would give all the children a dominant, recessive, and one in the middle. So I made a big long chart about it. I didn't use the color names as I was figuring out because that was confusing to me. So I figured out what all the ratios could be. Excuse me, for an incomplete dominance, and I got these ones. So you'd have, so if you had 2 dominants, you'd get a 1 ratio. If you did, this double dominant with just the 1 recessive, you get that. BB, 2 little b's, you would get just that 1 ratio. Here's the heterozygous, which would give us the 4 different options. If it's complete dominance, this would be a 3-to-1 because it's an incomplete dominance. You get 1, 2, and 1. Coming down here, you get similar thing, just depending on if you get a big B or little b in this one, and then nothing over there. And so I tried to see which of these ratios would best fit some of them. And the biggest clue was this first one, the Palomino, Palomino, or I think that's the yellow. Yeah, yellow, yellow. You saw that they could either go to the brown or to the white in that ratio. And so I plugged that in. And because Palomino's the yellow, and that one was twice as much as the others. So it showed to me that, okay, so Palomino is the middle one, but which of these traits is the dominant? And which one of these was it that I found that proved? Because I put that brown was the dominant. Because that's 1-to-1. Chestnut cremello. Oh, it's right. It's right here. The chestnut cremello. Or sorry, no, not that one. Okay, which one's white? Cremello and chestnut. Or no, that one's not the right one. Cremello, cremello, chestnut, chestnut. Actually, now that I'm looking at it, I know I had the brown coat as earlier, but I actually don't think I can be able to tell for certain which one is actually the dominant. Because I'm looking at this again. And you see it may actually be the white that's dominant because yeah, it is. Well, actually, now that I think about it, it really doesn't matter because you're going to get the same results because there's not a dominant and an incomplete dominance because they're both going to affect it. So for my example, I did BB, the chestnut hair, which was the brown, and then the yellow, and then the white. But the more I'm thinking about it, it could actually go either way. It doesn't matter which one is considered the dominant, at least in this case, because they both have equal effect. And then just some fill in the blanks. Glycosol transferase, that's the one that the A and B blood type alleles are using, and it's a galactose molecule that's put on the red blood cells. Overdominance is the example given, such as the sickle cell anemia, where if you have sickle cell anemia, if you just have one of the genes for it, you get some immunity to malaria. But if you get both, you get more immunity to malaria, but then you also get sickle cell anemia. So overdominance is the one where you actually want a heterozygous, because then you get some advantage from the recessive without the negatives of the recessive. So you get a mixture of both, and that's what you'd want. Maternal effect is where the phenotype is not determined by the genotype, but rather the genotype of the mother. And this is because the mother cells do some of the development inside, I guess, the fetus. This is in snails, so I don't know snail fetus, if that's right. But some of the mother cells come in and they help do things, and those chromosomes are kind of implanted and used as blueprints on how to build the snail structure. But once the snail starts growing, it loses track of that DNA and it starts using its own. But the structure's already started to be formed, and it just continues the way it is. So that's the maternal effect, where the mother's genotype is what the child's phenotype is, even though it may have a different genotype to begin with. Epistasis, that's the one where one gene, I guess it says right here, masks the phenotype effects of alleles on a second gene. And I'm trying to remember which one that was. Oh, okay. So that was the rooster head, where there are the two options that you can have for what type of, what's it called? Comb? I think it's called rooster comb. You know, basically the red thing on top of its head. There's two genes that help determine it. And if you have both dominant of one of them, it'll make a fourth option entirely. So that's what that one is. Phenylketonuria, phenylketonuria. There we go. So yeah, it's talking about how it's being modified by the person's diet. This is just saying if you don't have phenylalanine, one of the amino acids in your diet, that then if you have an inability to digest those and absorb those, that then you don't have to worry about it because you're not noticing that defect. This one I wasn't sure of. The Manx trait is kind of a spinal defect in cats. And it gives this really weird 2-to-1 phenotype ratio, which seems really weird, but the reason is if you get two of the dominant Manx traits in there, because it is a dominant gene, that it actually kills the fetus before it can, develop or mature at all. So of surviving offspring, the ratio will be 2-to-1, although in actuality it will maintain that 3-to-1. We just can't observe that by looking at the baby cats that are hanging around. So this one is talking about the maternal effect. It's the snails, the L. peregrina. So it's asking if you have a percentage such and such for the moms, and they randomly mate with males that are random. What are the phonetic type ratios of the progeny cells? And it's just going to follow what the mom's genotype was. So the moms have these following genotypes. And so effectively, as long as there is one dominant in the mother, the child will have the dextral, or right-handed, I think it was. And then there's 40% with the other one, which is the sinistral. I think it's left-handed, which is opposite. And so that's how you figure out that one. This is talking about the jalapa plants. And it's the same thing where the mother's genotype determines the child's phenotype. And so 40% are green and 60% are white. So that's what the children are going to be for the phenotype. And then if we come down to this guinea pig one, it's really just kind of the same Punnett squares we've been doing before and the ratios. It just changes a bunch with each color. And so you have to really think about this order where it's black, sepia, cream, and white. To determine dominance. So if you have offspring that are 50 black and 50 sepia, well, you know that there has to be a black in there. And so that's where you get one of the blacks, and then you have to have at least 2 sepias. I guess you could have maybe just one. Um, yeah, so you could actually have a few different ones. Uh, like, in fact, one I'm thinking of right now is you could also have or BS, which is going to be black phenotype, and you could cross that by a CC or white. Because then no matter what, black is going to beat everything, but this S, that could beat either of those. So let me just fill it out because I want to be complete. Phenotype X. It's going to be cream. I have to write phenotype every time. And white. Yeah, it's just basically a question on what is this parent giving. If it's black, it's going to be black. If it's S is going to beat out anything else the other parent can have if it doesn't have black. And so it's how you're going to get that ratio. Any of those will work. For this one, you have 75% sepia, 25% cream. So to get that 3 to 4, they need to be heterozygous, S and C. So that means if you get S here, then it doesn't matter. You're going to get at least half of them sepia. And then over here, if you get the S there, or C and an S, you're going to get another sepia. But then both C's will give you that 25% cream. Come down to C, so we have black, sepia, and white. So it means that we need to have white in both parents, because if we don't, there's not going to be a way to get just that recessive to be alone. So it's how you get the W and the W. And then black and sepia are just going to be put in there. So then, if black goes with white, black wins. If sepia goes with white, sepia wins. But black holds a tiebreaker, and that's what raises it up to the 50% from the 25. And then if we have 75% black and 25% white, just similar to up here, it's going to be that 3 to 1 ratio. So you just need 2 blacks phenotypes, both with a big B, but they also need a white to be able to allow them to get that white offspring off of there. So that is all the questions. So thank you, and here you go.